Okay, good afternoon and welcome to the Simon's Electron Microscopy Center's Winter EM course. A couple operational notes. So there's been a scheduling conflict with our next speakers. Our next section is about 2D crystallography. Aban Uberachena is supposed to speak March 27th, and now that's shifted to April 5th. So for those taking the course for credit, we'll live stream that and we'll record that in case you can't come. Hernando Sosa is still scheduled to talk about helical April 3rd. So again, the next section after this section is about 2D crystallography and helical reconstruction. The March 27th lecture is now going to be on April 5th, and the April 3rd lecture is still on April 3rd. And that sort of brings up our current topic today, single particle analysis, the last of the three lectures. And our speaker today is Rich Hite, a faculty member at Memorial Sloan-Kettering. He had a previous life as a 2D crystallographer, and now he's grown to love single particle analysis. He's very proficient. And as I mentioned, all our single particle lecturers have been cross-trained in many different techniques, but they have certain specialties. And I would say transporters and channels, memory proteins in general, is one of Rich's specialties. So if you have questions about your particular project, because I know a lot of people are interested in working on memory proteins, you can see him after. Okay, so without further ado, I'm going to introduce you to Rich.
Hi everyone, thanks for coming. So I'm going to present what I hope is the third of three single particle lectures. I hope there's some continuity with the previous lectures. I looked at their seminars and spoke with them briefly about what they were going to present. So I hope there's not too much overlap. If there is, let me know and I'll try to use a slightly different viewpoint to present something that you haven't necessarily already heard before. And so the overall goal of what I want to present is, what do you do once you have your density map? The previous lectures I think were about how to set up the microscope, how to collect data, what are some of the various approaches that you can use. But now, once you've got a map, what are you going to do with it? How do you interpret it? What does it actually tell you? What can you really tell from it? And what do you not have the information to be able to interpret that? And so, please ask questions throughout. It's much better, I think, in these sort of lectures if it's interactive rather than me just talking at you. And so this was, a few weeks ago when I was starting to prepare the lecture, I went into the EMDB and looked at the first map that was above 5 angstroms. And this was one that I pulled out. It's the apoptosome. And it actually, for going through what I want to present today, it turned out to be almost ideal test case. There's a lot of interesting features about it, as well as the way they processed it, what they were able to interpret at different levels from the map. And so my thought process whenever I get a map is starting out, how do you validate your map? What evidence do you have that actually supports the hypothesis that your map can faithfully reconstruct the structure that you have? What do you have that can tell you that, yes, this is really what I have, versus I just threw some voxels together, or it's entirely modeled bias, or any of the number of things that we know can go wrong and have go wrong with single particle analysis using cryo-electron microscopy. And then following up on that, what methods can you use to provide additional evidence to support your hypothesis that your map does represent the protein that you've put into the grid? Next is, once you actually now believe that your map, yes, does represent the protein that you think you have, how can you interpret it? So people always talk about resolution. Resolution is, in many ways, the most important and the least important feature about cryo-EM. So more important than the number is, what is the resolution of the features of your map? So if you think you have a 4 angstrom map, do you have features that represent that? Do you have side chains? Do you have good separation of your beta strands? And that may not even be global. That may be some region, and whereas other regions have much lower resolution. And so whatever region that you want to discuss, whatever part of the structure that you think is important to tell your biological story, make sure that the features in that region actually represent the data that you want to present. And then following up on that is, how uniform is the resolution? If you have a 4 angstrom map and some parts of it are really good, you can learn a lot more about those resolution-wise, and so the detail that you can see, than other regions. And that's why this map, I think, will be useful as a test case as we go through. And then the last thing, and I think the most important thing, and one of the real strengths of cryo-EM, but one that most people tend to forget, is sample heterogeneity. All of the samples that we put into the microscope have some level of heterogeneity. And how heterogeneous that is can either be informative or it can be a real disaster when you're trying to do a reconstruction. And trying to balance the amount of information you can get from your heterogeneity with the ability to actually get information from the sample is a really important thing in single particle cryo-EM. Any questions? And so the first thing, and I think the most important thing, and one that now that people have direct detectors and everyone's getting high resolution kind of gets lost, is how to validate a map. It used to be that there was a whole set of criterion that every journal would require you to go through before they'd allow you to publish it. Now, if you see side chains, they allow you to just kind of ignore it, which, looking back at crystallography, just the presence of side chains doesn't necessarily mean that you're right, nor that you've done a good job in reconstructing your molecule. And so the first thing is, what is the effect of initial model bias? And so as you all know, we start with an initial model, and we use that to generate our higher resolution reconstructions. And one of the things that most people forget, but a few years ago was a real issue and came up over and over again, was whatever you put in, you're going to get back. So if you put in a 3 angstrom reconstruction and then you go through all of your processing steps, you're going to get a 3 angstrom reconstruction out regardless of how good your data is. You could put total white noise data in and get that same reconstruction. And so this is a really important thing to be careful of, is whatever you put in, you know you're going to get out. So any information that you want to get out has to be beyond that. It has to be significantly beyond it. This also applies to masks. Previously, or more recently, people have been using masks extensively doing cryo-EM reconstructions. Sometimes it's to look at a particular domain, sometimes it's to remove floppy pieces, but the shape of the mask will very strongly bias everything inside of it. If you do the autocorrelation of white noise and a white noise mask and look at it, you'll actually see that there's a lot of information that is maintained just by applying that mask. And because cryo-EM is an iterative process, using the mask over and over and over again, you put a very strong amount of constraints on the reconstruction that you can end up with. This is one of the reasons why you have to be very careful when using masks, and also that when you use a mask, you should make sure you can get almost an identical reconstruction without using the mask. And then perhaps the most famous and the most important one to think about is this Einstein from noise theory. So if this was something that came out with the HIV trimer about, what was that, 4 or 5 years ago, Ed?
Yeah.
Where there was a group that had a reconstruction and they had data that they said supported a 6 angstrom map or whatever, I don't remember the details, but they had used a very tight mask and had used a very high resolution model to do the search from. And so they started, I think, with 6 million particles or some crazy high number, and were able to get back a 6 angstrom reconstruction, in which they said there was information to 3.5 angstroms. But when anyone else took their data and used and got rid of any of the masks and got rid of any of the high resolution initial models, they weren't able to get anything past about 20 angstroms, indicating that they had, the group that had published this structure had forced some of their data through the use of initial models and the use of masks to look like what they wanted it to look like, rather than what the data actually said. And so this is a reconstruction of Einstein's face from a whole stack of data where they just use white noise. And so this is something that you always need to be careful with. And when you're evaluating other people's work, you need to quickly look in the methods and make sure, how did they get their map? What are the masks that they applied? What is the initial model they applied? And so does their data actually demonstrate what they think it does? Any questions? Anyone had any problems with this, encountered this during their work?
Actually, I have a question. At what stages can you apply masks? Can you apply them like at almost any step along the way, or?
So that's one thing that's I think still up for debate. You can apply a mask at any stage, and if you justify it and you also do it in parallel without the mask, any results that you obtain with the mask should be representative of what you'd have without the mask. But the mask is some ways going to help you, but in other ways it's going to hurt you. So you have to be careful that if you can get a 3.6 angstrom reconstruction with the mask, can you get a 4 angstrom reconstruction without the mask? And is there a reason when you look at the map to think why the reconstruction would be worse without the map? Are you getting rid of floppy regions? Are you getting rid of regions that you know may or may not be because of sample heterogeneity? I think masking during particle picking is a real gray area. That's one where you probably want to be really careful, because if you mask out white noise, if you put Einstein's face and just put a mask, you're going to get it back really easily. And so there you're going to have a lot more constraints. But once you've picked your particles and you have an initial map, then it's easy to run through in parallel with and without. Because you've already picked your particles, if you pick them with the mask to begin with, then you're going to bias things much more strongly.
Also depends on terminology. Is your mask just a spherical mask, or are you making a highly detailed mask?
Right. But even a spherical mask will introduce correlations in your map. No one ever talks about that because it's one of the reasons why cryo-EM works. If you take away your spherical mask and do a reconstruction, processing takes forever and sometimes doesn't converge at all, whereas if you apply a spherical mask, it converges quite nicely. Particularly now that people are getting to higher resolution and have to use larger and larger box sizes. The calculations are greatly sped up by using a spherical mask, and so that's why every program has it as a default. But it does introduce correlations that are not present just in the data itself. And so, getting back to initial models, one thing that we always think of, we kind of ignore, is that we get an initial model and then we get a map from it. But if your map isn't necessarily 2.5, 3.5 angstroms, where you can see extremely high resolution detail, you need to be careful that your initial model does represent the data that you have. And so one way to test this is to try 3 or 4 initial model programs. There's quite a few now, and they're getting better and better, and they seem to work faster. So it shouldn't be that cumbersome to take a small subset of your data, run them through a few initial model programs, and see if you get similar looking reconstructions with all of them. The next is, a lot of us, I know in my lab, we often work on molecules with 4-fold or higher symmetry. And some proteins have even higher symmetry. Viruses, Ed last week was working on something with
24-fold symmetry.
24-fold symmetry. And so their initial models take a lot of that into account when generating it, and that can quickly speed up the process of convergence. But you want to make sure that your map looks very similar without imposing the symmetry. Make sure that that symmetry is properly imposed. Sometimes you have a subset of your particles that actually have the correct symmetry, but then there's a large number of particles within the data set that don't obey that symmetry. Either a subunit's lost, or there's conformational heterogeneity such that some of the particles obey symmetry throughout, and other ones it's only some fraction of them. And with membrane proteins, there's now been a lot of structures recently where they've looked at what you would think would be an overall symmetric channel, but really in parts it does obey symmetry, but in other parts it completely loses the symmetry, and they think this might be biologically meaningful. And so then again, when you, not only for initial model, but for your reconstruction itself, is how did you define the symmetry of the sample? Sometimes low resolution symmetry looks to be very apparent, but as you get to higher and higher resolution, you see that that symmetry may not actually be truly justified, and that there's going to be differences between subunits in your sample. And so it's always important to generate a reconstruction without applying symmetry and look at how similar that compares. Another way that you can test this is if you think that you have a loss of symmetry, is rather than doing a reconstruction of your full molecule, is if you know what the symmetry operators are, and you have, say, a 4-fold symmetric molecule, you can rotate it 90 degrees, cut out each monomer, and then do a reconstruction of those, and then do classification on that. And that will let you know that sometimes even within a molecule you may have 2 subunits that are the same and 2 that are different. By doing classification on individual monomers, because it's not always going to be the same subunit. Sometimes it can be diagonal, and sometimes it can be pairwise. They're going to be in different conformations. This will give you the opportunity to sort out different particles that have slightly different conformations within them, but not necessarily throughout the entire molecule. And one last thing that can be particularly powerful and is often neglected, and something that crystallographers have been using for decades, is internal or pseudo NCS symmetry. So oftentimes you have a complex, sometimes you may have 3 or 4 copies of one subunit, 2 copies of another, and 1 copy of another 3 or 4. But because those 3 or 4 copies of subunit A should be ostensibly the same, what you can often do is, if you can see where they are in the map, you can generate the coordinates for them, and you can generate parameters to move one to another, as you can average within the map those together. And that's a good way to validate. If they all look similar, then you've probably done a pretty good job of the reconstruction. However, if they all look different, and you don't have any explanation for why they're in such different states, then you might have some mistake that's been made. Or you're going to learn some biology about why different subunits in a complex are going to obey very different conformational states. And so that's what I was going to say here, is just you can use averaging. And so recently, as I just mentioned, there's been a lot of ways to generate initial models from your data. And one that just came out a few months ago is this cryospark technique. And what this does, which is kind of nice, is it doesn't force you to do 2D classification and pick only the classes that you like prior to generating your initial model. In this technique, you give it a full data set, and it comes up with some random initialized model. Most likely looks nothing like your reconstruction should, but then it subsamples the data over and over again and tries to generate things that look like itself. And you can either tell it that you have one initial model, or maybe if you know that within your data you have 3 or 4 different types of conformations, and you want to try to sort those out at the initial model stage. And so you can tell it that you have 3 or 4 different samples that are all within this one data set, and you can start to sort those out. And so by doing this, you start with just initial raw particles. By subsampling over and over again, you can start to build up information about what the molecule should look like, and then you can get a pretty viable initial model. This can then be put into Reliant, Freeline, whatever it is your reconstruction program is that you like, and then go to higher and higher resolution. Other programs require you to do initial 2D classification, but the problem with that is prior to putting those into your 3-dimensional reconstruction algorithm, you have to select which of the classes you like. Which views do you think look good? Which views make sense? And if you know a lot about your molecule, this can be good because you can get rid of things that are clearly junk. But if it's a totally new structure that you don't have any information about, oftentimes you're going to select the particles that you want, and you're going to get rid of things that you don't want. And so this may bias you only to have a certain set of views, and it can really screw up your initial reconstruction. Have any of you done initial model reconstructions? What did you use?
Cryospark.
How did it work for you?
Actually really well.
Did you try it with just one initial model or with several?
The protein that I'm working with is really flexible, so there's a lot of heterogeneity, so I had to make a couple of models.
And were they different? And did they represent things that you think might represent the channel?
Yeah, I think so. Some of the data set that I was working with wasn't terribly clean, so some of the initial models came back as kind of junk. It looked like a lot of noise, but a few of them certainly looked like what I expected, with some slight differences that were pretty interesting. So.
Oh, there's another vote for Cryospark. Has anyone tried any of the other ones?
It's also really fast. Which
Yeah,
is great.
it's nice. You can set it up, come back in the morning, and have a handful of initial models. EMAN 2 is also extremely fast, but it's kind of hit or miss. Sometimes, particularly if you have symmetry, it'll work really nicely, and in a half hour you'll have an initial model. Other times, if you don't have any symmetry, you can just run it and run it and run it and end up with things that don't make any sense. And so there's a lot of programs to try. I always think it's best to try a handful, see what you can get from them, and then try your high resolution refinement. And if you can improve the initial model, then it probably means that it's right, or at least there's some evidence to suggest that it's right. And so another form of validation, which I think if your map is under about 4.5, 5 angstroms, should still be required for every reconstruction put into the EMDB, is tilt pair validation. And so what this does is it tries to give you an idea of how well you can align your particles. And so what you do is you start out with initial reconstruction, some number of data, and then at the end you just take a few images both at 0 and at some fixed tilt angle. It doesn't have to be 60 degrees, it doesn't have to be extremely high, it can be fairly low, although the work that the center has done here with tilted has been really successful at 40 or 45 degrees ahead.
As far as you can tell without losing resolution.
Right, which is really, really nice. And so this might become a really powerful way. Now you just do your last 10 images and you can do it at 40 degrees, and that will very quickly tell you how well you can align your particles. So what you do is you take your image of the same particle at 0 degrees and at some tilted angle, and then you go through and you calculate where every particle is in both images, run them through your alignment program so that you can get a set of angles, all of the different alignment angles. You then cross-correlate each of those two and look for the displacement vector. And so you draw a map that looks similar to a normally Euler angle plot. But what you can do is compare, if this is 0, so this is where all of your untilted particles are, you then look at all of your tilted particles and see where they start to cluster. If you've tilted your specimen to 20 degrees or 15 degrees, and you can see that there's a big cluster between 0 or between 10 and 20 degrees, then you've probably done a pretty good job. If they're totally scattered throughout the image, that indicates that you can't align your particles very well. One side effect of this is that the bigger your particle is, the easier it is to align, the tighter this should be. And so this was done prior to direct detectors. This was done either on CCD or film, I don't remember which, with beta-galactosidase, which at that time was still a fairly challenging specimen, not that big. Now people have done it to almost 2 angstrom resolution, and so I imagine that these plots, these would be much, much tighter. But using film or CCD, this is a pretty reasonable approach to going through and calculating how well you can align your individual particles within your data set. Any questions on this approach? Another one, and now is actually built into Reliant, and so I'm sure a lot of people have seen this in papers and have used it, maybe not even known it. If you use Reliant's particle polishing, or not particle polishing, the, what is it, Red?
Post-processing.
The post-processing step, it automatically includes this. And what this does is it tries to estimate the effect of the mask that you're applying during your reconstruction. And so if you run your entire reconstruction algorithm with a spherical mask, you end up calculating your FSC for not just the protein component, but all that space between the edge of your protein and the edge of the spherical mask. And so there's a lot of noise in there. Some of it will probably be negatively aligned or positively aligned. It doesn't really matter. It's not going to contribute positively to your reconstruction. You're actually going to end up with negative correlations due to it because you're trying to align noise where there isn't any signal. And so one way that you can try to actually look to see if you have a better alignment of the protein part with the protein part is to generate a mask that just includes your protein, so a fairly tight mask, and calculate the FSC between those. But as I said earlier, you get a lot of correlations just by having the mask included. And so this approach tries to calculate what are the effects of those correlations. And so the way they do that is you take your reconstructions, your 2 half maps, and you cross-correlate them. And then you take your reconstructions again, and at a certain resolution, usually somewhere around 10 or 8 angstroms, you put all of the phases beyond that resolution to noise. You just randomize them entirely. And so there you should have no correlations from data. And so any correlations that occur between your 2 half maps beyond that resolution are entirely due to overfitting or noise correlation. And so if you look at these different plots, in this one, they calculate here. And so this blue line represents random phases beyond 10 angstroms. And so you see once it gets to 10 angstroms, the correlations drop immediately almost to 0. So in this reconstruction, there's been very little overfitting. At 8 angstroms, it drops a little later, and then what you can do is using this formula where you calculate what they call FSC true, you can calculate your FSC data from your FSC noise, and between those two you can calculate this FSC true, and this represents the FSC from your data, not just due to the overmasking. And so this is this blue line here. And so what you have is a combination of this, where you have the randomized data. Here is the data where you've applied the mask, and it's somewhere in between. That's what your actual FSC is once you've taken into account the noise correlation. And depending on how well your program has done, how much over refinement you've had, this line can shift in different ways. And so if you have an extremely overfit map, which is this example here, you can see that beyond about 1/1.5, the random data and the real data are almost perfectly overlapping, indicating that almost all of your signal is due to overfitting of the noise rather than actual alignment of signal. And so when you run the calculation, you can see it ends up with a similar reconstruction, overall resolution of about 6.5. And you can see that no matter what mask they use, they all seem to have similar resolution, indicating that this is a mask-independent calculation, and the correlations you're getting are just due to how tight or how loose your mask is. Any questions? Has anyone tried this, knowingly or unknowingly?
Reconstruction programs have you been using? Reliant? Reliant? Reliant?
So you haven't been doing post-processing? It will give you a better estimation of what your resolution actually is, but it doesn't make sense if your reconstruction's lower than about 10 or 12 angstroms, because the effect of this mask becomes massive compared to the effect of what you're able to compare with the phase residuals. And so now that we've gotten what we think is a true resolution, what does it actually mean? And so one thing I always look at in my reconstructions, and I think is important to look at, is what is the actual resolution locally? So the overall resolution doesn't really have a whole lot to do with what local resolution is. In some cases, you can see all of the details you'd ever want in some region, but the region that you're actually interested in is entirely blurred out. And so your overall resolution doesn't apply to either of those pieces. One of them is going to be a much higher resolution, one of them is going to be lower resolution. And so it's really important to take a good amount of care to evaluate what the part of the map is that you want to talk about. What is the biological relevance, and what can you interpret from the resolution at the particular region that you're looking at? And so I think that local resolution estimations are extremely important. And so there's two main programs that can do this. One of them is ResMap, which is included as part of Reliant, and the other is BLocalRes, which sometimes does a slightly more accurate way. ResMap, particularly if you have a huge volume in a very small reconstruction, you can end up with values that are far beyond what really makes sense if you look at the density. BLocalRes is much slower, however, so sometimes it can be useful to look at both to try to get an idea of what your local resolution might be. But in the end, evaluating your map by hand is the most valuable way to get an idea of what the local resolution actually is. And so one of the ways is, if you have a big reconstruction and you don't want to go through and look every single side chain throughout the whole reconstruction, or if you just want to do a quick overview, is to use density slices. And so here again is this same reconstruction, but now rather than just looking at the density, we're looking at a top view and a side view. And you can see down the top, the center, there's extremely good resolution. But these peripheral domains are almost entirely washed out. And again, from the side, you can see that there's a top domain that I don't know if you could see it at all looking from the top view. And so this is quite common, that particularly if you have symmetry that's not obeyed by the entire molecule, is by applying symmetry, you're going to wash out domains entirely.
Can I ask, what do you think, what would you say that 4.4 represents then?
4.4 in this case represents a mask around this domain with symmetry opposed. By reading through the methods, I was able to discern that. But looking at the map overall, it could be very difficult to tell what local resolution might or might not be at any point in this map. And that's why using, in this case they used ResMap, you're able to come up with a quick graphical way for everyone to go in your supplement and say, okay, yeah, this part actually does have a higher resolution, so it's somewhere between 3 and 4.5, whereas these peripheral domains, as you'd expect just looking at the slices, are much more washed out, or somewhere in the 6 to 7.5 angstrom range. And so what you can do, what you can interpret, is really important. At 3.5 angstroms, depending on how much you know about the protein, you might be able to build a de novo model. At 6 angstroms, there's no way you're going to be able to build a de novo model. There you're going to be working on fitting in maybe homology models, or just trying to interpret where pieces of the protein are relative to one another, rather than looking at individual side chain interactions.
Just looking at this here, is the resolution a little worse in the dead center, just more flexibility there, you think?
Could be more flexibility. Sometimes, if you've ever done anything with symmetrization, you notice that there's a lot of noise along symmetry axes, because you're trying to force everything there to obey the same symmetry, and sometimes that's not necessarily the case. And so you'll often see right along the symmetry axis huge peaks, positive and negative. Any other questions?
So if you do the Fourier shell correlation and get a resolution, is it then more or less the average of the ResMap, or?
Depends on how you calculate your Fourier shell correlation. If you apply a mask just for this domain, you're going to get a Fourier shell correlation that is most likely somewhere around the mean of this domain, but not the mean of the whole thing. If you do your Fourier shell correlation around the entire thing with a spherical mask, that's probably going to be at the low end of your overall. But they're not necessarily mathematically equivalent. One of the differences is that what BLocalRes does is it takes little chunks of your density and does a Fourier cell correlation on that. It doesn't necessarily correct it with the mask though. So you can, and the same with ResMap, is because you're not applying a mask, you're just looking at local regions, you're going to end up with the same sorts of correlations that we talked about in the previous slide, where by looking only in a small region, you're going to end up with built-in correlations because you're eliminating all the other information around it. And so this goes back to what your question you asked. And so 4.4 is the apoptosome platform, and so this is putting a mask around it, calculating the Fourier shell correlation for that with symmetry. The central cards, so those are the first domain. Here you can see they have a reconstruction at 5.1, and then everything put together without the symmetrization is about 7 angstroms. And so depending on what part of the reconstruction you're looking at, you have a very different Fourier shell correlation, so the way that you can interpret it is quite different. But this is now, a few years ago no one had even thought of this, and now it's getting more and more precise what you can do with masks. Because we know that lots of different reconstructions have different regions that move independently, and you don't necessarily need to know what every piece is at high resolution together. Sometimes you can go through and calculate different pieces, get a higher resolution map of that, build the reconstruction there, and then put them back in. Because in this map, you're not going to be able to build a high resolution reconstruction of this, but say you use the approach that I was talking about, you use non-crystallographic symmetry averaging of each one of these particles, put them together. If you can then figure out what the individual orientations are, you might be able to build a higher resolution reconstruction of this domain overall, even though once you put it back in, you're not going to keep that resolution. But it'll help you to build that reconstruction. Does that make sense? Do people understand what I'm saying?
So you could make local conclusions regarding individual side chains, but only within that one particular context. You couldn't actually fit it into the overall.
Right.
Resolution.
That's where you have to be careful. Is say you have a lysine that would be sitting here that might form a salt bridge with this portion over there. Because you know this domain in different subunits is going to be in different positions, some fraction you might be able to have that salt bridge, some fraction might not have that salt bridge. And so that might be biologically relevant, but you're going to have to get additional data that is orthogonal that can help you make that conclusion. Does that make sense? And so, as I keep coming back to this, but I think it's the most important thing when evaluating at any reconstruction level is let your map be the guide about what you can interpret. Sometimes you have a good Fourier shell correlation, but when you're looking at it, everything just looks a little bit broken, and you can't quite understand how everything works together because your helices and your beta strands look good, but the loops between them are all broken apart or are very hard to trace, and so it can be really hard to do a full all-atom tracing of the reconstruction because you know where all the helices are, you know where all the beta strands are, so you can fit secondary structure into them. But until you get to even higher resolution, sometimes it can be hard to see how they connect and then thus register everything accurately. And this is a real issue in some maps, but people have been pretty careful, so there haven't been too many well-publicized mistakes made. And I think that continuing to use your density as the way that you interpret your map is the way to continue to avoid those problems from popping up. Okay, and then this is what happens so you've gone through all of this and you think, yeah, my map's at around 3.2, 3.3, it's really nice, Ed's happy, he thinks I've done a good job. The next thing is to try to go through and evaluate it locally. So if you have that sort of resolution, you clearly see side chains, the backbone's well done, then you can use COOT or O if you're old school and do a crystallographic model build into it. If you have a homology model, sometimes it can be really quick to go into Chimera, dock it in, and then just do a few side chain changes in COOT or O, and you'll quickly have a reconstruction. If you're a little bit lower, if you're at 3.6, 3.8, sometimes you may be able to build some parts, but other parts there's just not sufficient information. And so in this case, it can be really helpful to spend the time using some of the other programs that have been built. I know the group at Washington, David Baker and those guys with Rosetta have built great tools that can use predictive information from the PDB to try to help you to build into something that maybe on your own you wouldn't be able to do. You have to be careful though, is the program able to build it accurately? And so sometimes even when you use that, it may be useful to trim away a lot of the side chains and just leave the polyalanine model at the end rather than trying to overinterpret something that you don't have good data for. And when you get even a little bit lower, then you're probably just limited to docking of crystal structures, or if you have good homology models, those can be. But again, how good is your homology model? How good is the data that went into it? You don't necessarily want to use someone else's homology model in your reconstruction, because then you have to depend on them doing it right, and your interpretation of that relies on them doing it right, and you don't know exactly how they did it. So it might be worth learning a little bit about some of those programs and doing it yourself. And then this goes back to the point I just made about sidechain interactions, is even if your resolution is high, don't make too many, don't say things about salt bridges, don't say things about hydrogen bonds unless your density actually supports it. Arginines and lysines can go in a lot of different ways. They're very long side chains. And so just because you can see the start of the side chain of an arginine and the start of an aspartate doesn't necessarily mean they're going to be a salt bridge. They can diverge in a lot of different ways between where you can see density. So be very careful about over-interpreting side chain level interactions when you don't have side chain level information. And I think we'll take a little break here, and then I'll come back with heterogeneity after the break.
I was just wondering actually what kind of resolution they need to see a ligand small molecule.
It depends. Sometimes I've looked at maps that are 3.5 angstroms and ligands are great. Other times 3.5 angstroms you see no good density for the ligand. You know that there's something there, but there's no way that you could orient it or register it relative to the protein. Depends on what the occupancy is, depends on how flexible the ligand is, depends on how flexible everything is around it. Any other questions? Great.
5-minute break?
Can I get a cup?
Oh, yeah.
Because with Freeline, you can do it pretty reproducibly at the time.
Yes, that's funny.
So I don't know.
5 views, and you pick 3 years out of 5 years. So I got, I mean, if I finish the program in 5 years, I still got a year before the so I can take all 3 years of grades out of that for just my last year. Apply for the wage or something. So I can apply for that as an extremely period for a year, and I can actually look at back up later. So there's something flexible. I think next year I start looking for take on myself
How's the Tri-Institute program? Are you getting a lot of rotation students, or?
So I have one rotation student from the transitional program, and I've had 2 from the joint Cornell MSK one.
Oh, so there's a separate
There's all sorts of different layers. There's 3 different tri-institutional programs too, I think.
Really? But you're in the structural biology program, right?
It's the chemical biology.
Chemical biology, I got that wrong.
Yeah, but that one has the majority of the structural biology students. Yeah, so there's one in Drew's lab that was the first author of the human CFTR structure. And they have multiple confirmations now, so they have more papers that they're writing on that.
Okay. As soon as the zebrafish one came out, everyone was like, oh, you need to yes.
Yeah, they got the human one not too long after the zebrafish one came out, though the resolution was a little bit lower, so having the human one was helpful. Or the zebrafish one was helpful.
Yes. From what I saw, you briefly mentioned the new notable about this, like basically it's the same overall architecture. It helps because people were just putting in asterisks already, saying, oh, but the human's going to be different. Maybe not that different.
Yeah, they released the coordinates of that one before they even submitted the paper.
I think in the CFTR community, there's a couple. I'm a little bit confused, but there's like a big consortium, and there's a couple of different approaches. People have been going about it.
Yeah, people have been trying for a long time, but weren't able to get a structure.
That was the holy grail for some people.
But there's also
A lot of crystallographers are really slogging their way at that one.
Yeah. Do you have many people here that are working on ABC transporters?
Let's see. From Columbia, there are.
Actually, there's one lab.
Don't exactly know if it's necessarily ABC transporter. ATPases, that's a larger category.
Right.
There's quite a bit of interest on AAA ATPases with DNA helicases. I think that's more. There's only a small segment working on ABC transporter.
Yeah, they're pretty small.
People working on AAA ATPases. I want to see the DNA. Right? Everyone's back.
Okay, before I continue, is there any questions from the first section? Anybody have any comments? Anything that they think they don't agree with, they disagree with, they think was helpful? Anything I could expand upon? This is for you. I want to present this in a way that's helpful.
We already know Ian. It's mainly, what do you want to learn?
I don't know about that, but I know something.
A question about the comment you made about the ligand. So you're saying that in some cases, even at low resolution, you can see the ligand, and you're saying in other cases, you don't see the ligand until high resolution?
Other cases, even at high resolution, you won't necessarily see it. Ligand occupancy is, because we don't have a crystal, we're not forcing everything to obey one conformation. You often have a mix of samples. You'll have some that have ligand and some that don't. And ligands are often such a small fraction of your overall mass that it can be really hard to sort ligand bound versus ligand free out if there's not a larger conformational change accompanied by it.
I see. That's why you end up seeing those observations. Because you've actually incorporated particles without the ligand into your...
Yeah.
I see.
Yeah, and it can be really hard to sort those out, and there's some circumstances. Other times, if your ligand generates a 45-degree rotation or another large conformational change, then it's quite easy, because just your global particle classifications should sort out the ones that are ligand-bound from the ones that are ligand-free.
How often have you seen ordered water in your channel structures?
I haven't seen it. None that I would believe, but I haven't gotten beyond about 3.4 angstroms, so.
And these sort of again, so the issue with the ligand there is because some particles aren't capable of binding ligand, or is it because the affinity was so low that at the experimental conditions you don't have full occupancy of that molecules?
Could be a mix of those two. Even a lot of times with saturating ligand, you don't necessarily have 100% occupancy because you're always at equilibrium. Your equilibrium, the more ligand you have, you're going to shift it further and further, and you may drive it nearly to completion. But some things don't get driven to completion. Other times, ligands will bind but not necessarily generate the conformational change, because that's also an equilibrium interaction. So there's multiple equilibria that you have to sum together to get things that are in conformation plus the ligand bound.
Okay.
And so that is quite helpful, because now we're going to talk about heterogeneity. And so within cryo-EM specimen and within all biological specimen, there's going to be multiple types of heterogeneity. I think for the types of specimen that we tend to work with in single particle, and even for some types of tomography and 2D crystallography, there's going to still be just these two main types. And the first is structural heterogeneity. And so this is, you have conformations that are different. You have pieces of your same complex that are in different places in different complexes in different samples. The other is compositional heterogeneity. So you have normally an 11 component complex. Sometimes you're going to have 8, sometimes you're going to have 10, sometimes you're going to have all 11. And so if you go through and average everything together, you're going to end up with portions of the reconstruction that are going to be very weak, because that component is only there in some fraction of the particles. And so this is the same as ligands. You can think of ligands as just another component in the complex. The higher the occupancy they are in your map, the better you're going to be able to define them. The lower the occupancy they are, the less well you'll be able to see them, and the harder it is to be able to do any interpretation when you finish them. And these, as I was just saying, will degrade the resolution of the region that they're bound to and the overall resolution, because you're going to be averaging things that are different, and so your particle alignment algorithm is going to have a harder time forcing things to look similar. So there's a couple different ways to overcome heterogeneity. But briefly I'll talk about this. And so this was one of the groundbreaking papers from Venki Ramakrishnan's lab a few years ago about ribosomes, and how they were able to take a total mess of what, even when they started working on it, they thought this would be a disaster. There's no way we can do it, because what they wanted to look at was the structure of the mitochondrial ribosome. And there's no way that they could come up with where they could separate mitochondrial ribosomes from cytoplasmic ribosomes. And so in the end, they had a sample that was heterogeneous. There was no way around it, they said. They couldn't come up with a biochemical purification where they had a reasonable amount of both specimens at the end. And so what they did was they just dealt with it. They threw it all into Reliant and then tried to use particle sorting to sort out what was cytoplasmic and what was mitochondrial. And so that's what this first step was, is they started out with an initial model in about 100,000 particles, and they ended up with the majority belonging to these classes, which they thought looked like the mitochondria, and then some particles that were cytoplasmic and some that were overall junk. And then, because they not only had not only had heterogeneity because they had two different types of ribosomes, they also had different additional components. And so this is what they're showing as the mask for the large subunit, and you can see there's different amounts of density outside of that mask. And so by putting a mask around it, they were able to ignore everything outside of it, just look at what was inside of the mask for the large subunit, and build a reconstruction of that. And so they had some bad particles, but the majority were these good particles. And so from a relatively small particle population, they're able to sort out a lot of junk and get a very high resolution reconstruction. But generally, you don't want to have to do that. You don't want to force the computer to do sorting. Doesn't always work as well. Ribosomes are great because they're huge and they have a lot of phosphate, so they scatter well. But a lot of the samples that we work with aren't going to be that big and aren't going to sort that well. And so we're going to go back more to biochemistry. We're going to try to do this at the bench before we get to the EM. And so this is where I think most work can be done to improve a sample. If you have a poorly rebuilt model, your reconstruction is not getting very good, it's usually because your biochemistry is not yet ideal, and there's a few ways to optimize it. And so most of it is just spending more time with your biochemistry, trying to improve sample purification, and this can really reduce your heterogeneity. If you have a lot of bands on your gel and you're trying to look at it in EM, you know you're going to have a mess. If you only have one band on your gel, then it's going to be easier. But sometimes if you're looking at a complex, switching tags from one component to another can make a huge difference in the yield and in the purity. If you have a 6-component complex and someone gave you a construct that was tagged in one component, that may or may not be the best, but if you're having trouble, it may be worth it to go back and try tagging some of the other subunits, starting the purification from scratch, and see if you can get a cleaner purification in the end or a higher yield. The advantage of having higher yield is that you're going to have other components are going to be easier to get rid of, because you can be harsher with your washing steps, you can add an ion exchange step, you can do some of the things that you can't do when you have an extremely low yield purification. Did you have a question?
No.
And so other things you can do are just changing your purification, adding ligands, trying different pH, trying different salt concentration. Ligands tend to minimize heterogeneity of the sample, and so by having them in your purification throughout, sometimes that can be really helpful. Something like ATP is not really viable to have in every buffer all the way through, but maybe you can have it in one of the little tiny gel filtration columns at the end. This can help you to sometimes eliminate extra heterogeneity in the last step right before you freeze it. That way, instead of needing a liter of 5 millimolar ATP, which most of us probably can't afford, you need maybe 10 mils of 5 millimolar ATP, and that's something that's more likely to happen. The other thing is to go back and do construct alterations. This is something that crystallographers have spent a lot of time on, and for their samples, because they need to form a crystal, they spend a lot of time, and sometimes this is essential. You need to cut and paste and do a lot of piecing together to get a viable construct. But with EM, we try to get the most native conformation possible. And so sometimes using a pair of this, get a lower resolution reconstruction of the entire construct without doing too much molecular biology. But then if you know there's a region that's always flexible and is sometimes falling off, sometimes being a real problem, then you can cut that, or you can trim it down to try to eliminate some of the flexible loops, and that may give you a higher resolution reconstruction. But because you have the full reconstruction map, you can now use the two to make a better understanding of what it is, and you have good validation that your reconstruction does faithfully represent the overall structure. Another thing that people still do, and was very popular for a while but is a little bit less popular, is chemical cross-linking. If you have a lot of arms or whatever it is that are very flexible in your reconstruction, and you're having a hard time getting a nice core particle to work with, sometimes doing a glutaraldehyde cross-linking experiment can be quite helpful. There's graphics and a few other experimental procedures that people have developed that in some cases work extremely nice. In some cases you end up with a really non-native reconstruction. And so that's the one worry about using chemical cross-links, is depending on what concentration of the chemical cross-link reagent, glutaraldehyde or whatever it may be, sometimes you get back something that looks very much like your native reconstruction. Other times the cross-links force things that really have no business being in contact with each other, force them together, and you end up with just a ball that doesn't actually represent your reconstruction at all. And so even if you do need to do, or if you do the cross-linking, you should do a non-cross-linked reconstruction. Even if it's much lower resolution, it can help you to validate that the chemically cross-linked reconstruction does represent your structure. Okay, and so this is a slide from a review, or a methods paper that Shores wrote last year. And so Shores is the author of Relion. And so this is one of the most complicated reconstruction procedures that I've ever seen, but it works great. And so these are some of the various tools that have been developed. I'm not going to go through all of it, because it's every single sample you're going to need to use a different set of tools. But I'll briefly go through the idea of how they're able to take something that was so flexible and with 500,000 particles end up with all these different reconstructions for different portions, and this is a spliceosomal construct. And so they start out with an initial model, and they have 470,000 particles, and after 2D classification, they throw away about 20,000, 30,000. So the majority of their particles, from what they picked to the 2D classification stage, seem to be viable particles. The next stage, with 3D classification, they throw away about 2/3 of their data, so they end up with only about 140,000 particles. What this means is not always clear. Some cases it's because the particles are damaged during purification. In a very multi-component complex such as this, that may mean that you're getting rid of particles that are missing domains. that may or may not help you in your reconstruction when you go through and do this masking afterwards. But it's always a good idea that when you do 3D classification and you lose more than half of your data, try to think about why that data is getting thrown away. What about it is not positively contributing to your reconstruction? What about it is different than the particles that you want to look at? And then at the end, if you make a biological conclusion based on 0.4% of your data, how meaningful is that? How representative of that is of the full data set? And these are things that are starting to be addressed in cryo-EM papers, but it's still something that you always need to think about when you're going through your algorithm. Is it because you did a really terrible job of freezing and so a lot of particles get stuck at the air-water interface? Maybe it's just your sample is not very well behaved, and in the 5 minutes it takes from gel filtration to freezing, a lot of it crashes out or looks terrible. But these are things that you need to think about and that you should evaluate every time you do a 3D classification. If it varies hugely from sample to sample, of the or purification to purification of the same sample, then you might want to think about what happened on the day when I only lost 10% at 3D classification versus the day when I lost 75%. Did I do something differently? Did I add DTT? Did I take DTT away? Did I end up with a lot of disulfide breaking? There's a lot of things that can cause proteins to become unstable, and you can learn a lot of information by evaluating your 3D classifications. And so in this case, they went from 440,000 to 140,000, and this gave an overall reconstruction without masking of about 3.7 angstroms. But because there were domains in this that they couldn't interpret, they couldn't evaluate properly because of flexibility, they knew that the overall reconstruction said 3.7, but this was insufficient to evaluate large portions of the molecule. And so they went through some of these masking approaches that I've mentioned to try to see if they can get a better idea of what the protein is. And so this is, you can see up here, this arm, which they highlight here, is really poorly ordered in this overall reconstruction. And so what they did in this part is they did particle subtraction. This is probably the most complex way to do 3D classification, where what you do is you can take, instead of taking your particle that has supposedly data from every part of it, is you start out and you do projection matching and you get rid of the portions of the particle, not just from your initial model, but from the raw particle images. You can go through and calculate with cross correlation what view of the particle you have, and say you only want to look at the arm, but the arm is just maybe 10% of the density mass, is you can calculate a mask that corresponds to these, apply those to the initial particles, so they don't have particles here, but you go back to your initial particle stack, delete everything that isn't that doesn't correspond to the arm, and then go through and do your classification. And this can be really helpful because it greatly increases the signal to noise. Normally your reconstruction is going to be dominated by this portion. The arm is 10% of the mass. You're never going to be able to classify it well if all that other stuff is there. The best example of this, this is one of the best examples where they're able to take this very small portion, and by doing mask refinement, they can get high resolution reconstructions of individual domains because they eliminated, using this particle subtraction, everything outside of that. This is where they're able to take this little bit of the arm, calculate that, and they can see already with the initial 3D classification that there's a lot of different classifications. And so what they wanted to do to try to build up the signal, because this arm is only 10%, to see if there's any correlations. Does movement of the arm correlate with movement of the body? And can this help you to build a better reconstruction of the arm itself? And so they went through multiple rounds. They first classified just on the arm, and then they did classification on the arm and the body, and you can see how the arm is moving even in just this smaller portion. And they're able to get better reconstructions. And you can see within this, the strongest resolution is going to be the body, whereas the arm is going to be weaker. The other thing they did was to try to look at global conformational changes. And so here, rather than using masking and particle subtraction, they just went through with finer and finer angular sampling. If you've used Reliant, the default parameters for 3D classification are 7.5 degrees. And so if you have a very subtle conformational change, you may not be able to sort that out. And so you can manually set it to 3.75, 1.875, or if you really want to kill your computers, 0.09375, whatever it is, and run your 3D classification that way, where you're sampling an enormous number of very subtle very subtle angular differences but those may be able to pull out the conformational differences that you need. And so with this, they were able to calculate a reconstruction where they had a closed portion here versus an open portion here. Any questions about this extremely complicated reconstruction technique?
Usually when we start classification, in this the original model had 3.7 angstrom, and it improves later when, but why it's like initial model has higher resolution and others have lower resolution.
This is one of the issues about thinking about resolution overall. So this is going to have the most particles. This is going to be 140,000 particles, and so the portions here that are well aligned are very well aligned, and so you're going to get a very high signal in those domains. And because the cutoff is 0.0143, you can have quite a bit of noise and still be above that threshold because it's going to be dominated by this core domain. This part out here isn't going to contribute much to the Fourier shell correlation. But as you get to smaller and smaller particle sets, particularly here when you've done your masked and then your particle subtraction, you're only looking at a very small portion of the overall reconstruction. And so here you're going to have smaller FSC curves because you have one, fewer particles, but two, you've gotten rid of a lot of the rest of the mass that helps you to align your particles. You can't align one small domain as well as you can a big particle. Any other questions?
One more question, at what level we talk about reproducibility? In this case it's open and closed, but in crystallography sometimes there is alternate conformation within the same residue. In cryo, can that also be considered as heterogeneity?
It depends on how well, if you can sort it out. If you end up, when you look in a map of a crystal structure, often you'll see side chains in two different rotamers. I don't know of any EM structure where they've been able to do partial occupancy for side chains, but if it's occurring in a crystal, it's most likely going to occur in EM. It's just how well can you sort out. If you're looking at one side chain, so if you have 1,000 residue reconstruction and you're looking at the side chain of one residue, it's going to be really hard to classify that out. But if that side chain rotamer difference correlates with a larger conformational change, then it'd be pretty easy to sort it out. It's how much signal are you going to get between different conformations is the biggest thing.
Okay, so means we cannot go very too deeply this side chain level.
Sometimes classification can work at the side chain level. It depends on how fixed everything else is, and so how tight of a mask you can put on it. If you're working with lysozyme and every other residue is entirely exactly the same other than one side chain that's flipping between two conformations, you might be able to mask everything else around it and just do classification on that one side chain. I don't know if anyone's done this. I don't know if the signal to noise is high enough or if you can align particles that well, but that's the idea, is how much can you get rid of your background? And that's why this focus classification using masked particles can be really powerful, because you're getting rid of things that you think aren't going to contribute to the alignment. What's most interesting, though, when you do this sort of thing, is you actually find correlations, that small movements in the body correlate with large movements of the foot or vice versa. That would give you an indication that there is some coupling allosterically between the domains of the reconstruction.
Can you estimate the percentage of each conformation that you have?
So that's something that people are now starting to do, is look at, you start out, one of the problems here though is you've already thrown away 300,000 particles. What class do those belong to? And so when you're doing a very stringent 3D classification prior to doing this, then you're only looking at a subset. And what is that? And that subset could be all pushed to one extreme or all pushed to the other extreme, or it could be randomly sampled in the middle, and so you can lose information. But if you're able to get a really clean data set where you don't need to do too much 3D classification, then you should be able to reasonably calculate 90% are in conformation A, 5% are in conformation B, and 5% are in some conformation that you can't well classify. And so that would kind of be your error bar. And if you can do it multiple times, or you can subsample your data multiple times, then you can start to get a pretty good idea of this. And so this was an experiment that I did as a postdoc, was looking at an ion channel. It was a ligand-gated ion channel, and we calculated reconstructions in the presence of different ligand concentrations. So in the absence of ligand, it was all in the closed conformation. In the presence of high concentrations of ligand, the equilibrium was shifted very far to the open conformation, but in between, we were able to get a dose-response curve that looked similar to what we could get through electrophysiological assays, that the half maximal concentration was similar for both methods. And so I think a lot of people are going to continue to do this, add ATP at sub-saturating concentrations if you have an ATPase or something that's ATP conformationally generated, and look at how, because for decades we've been doing assays where you add something and you look at some sort of response, but now with EM, because heterogeneity isn't necessarily a downside, with crystallography, if you had multiple conformations, you just had a bad crystal. Here, now you can have multiple conformations in your EM grid, and if you can sample them well and you can actually characterize the majority of the particles, not just a fraction of them, then you can start to make biological conclusions based on the particle distributions at different ligand states.
Actually, related to that, is there any sort of metric for like me as a reader of a paper to say this person picked a representative selection of particles from the grid to start with? That like all of one conformation wasn't crap, you just didn't pick in the first place.
So one way that, depending on how big the conformational differences are, if they're using auto-picking, there's usually a low-pass filter that you apply to the reference, to the templates, and to the images. So with Reliant, I generally use 25 angstrom low-pass filter. And so often if you have a subtle conformational change, the difference between that isn't going to be that big. The other thing is, I don't know how many of you have used various auto-picking. Freeline, when they do it, it's just a blob. Reliant, I think, now has a blob search as well, where anything that is black, so anything that looks like a particle, should get picked, and everything that's not isn't. The problem is that means you're also going to pick any ice contamination, and so sometimes it can be helpful to go back and manually clean it, but then, as you said, you're introducing bias by manually cleaning it. And so this is an area that because picking algorithms aren't perfect, it can be helpful to try to reprocess it, have someone else do it, see if they end up with similar conclusions. That's really cumbersome and a lot of time, so I don't think anyone's willing to do that yet. But as the techniques get more and more advanced, hopefully the picking algorithms will get better and there will be less bias introduced. What sort of auto-picking or manually picking have people tried here? Auto-picking?
Yep.
With templates or with blobs?
With templates.
Did you pick the templates yourself?
Yeah.
And how did that work? If you went through an image, if say there was 200 particles, what fraction of them do you think it picked that were real?
Well, just to be clear, so I picked manually, like totally manually generated templates and then used those templates to do auto picking.
But say after you run your auto picking, say you have 100 particles in your field of view, how many do you think it would pick that were real? How many do you think it would pick that were false? And how much time did you go through cleaning it up?
It's a good question. I guess it's really kind of varied greatly with each data set. Of course, with the contrast, I think has been a big thing. It's changed, and like you said, it picks. If there's any gold or anything like this, if you're using a gold, using a gold grid, it'll pick on that automatically. So I have had to go through each my graph manually and just clean it up, but I don't do so. I don't unpick anything that may be a particle. I only unpick it if it's the edge of the grid or something like this. That takes forever.
Yeah. Yeah, I think right now cleaning up picking is the slowest part of the procedure, and so if there can be auto picking works great in some cases if you have a really clean grid. If you have something that's aggregation prone, or you have DNA which can just cause beads on a string sort of thing, then it can be a real mess. And so as a reader, looking at the individual images, particularly if they can show you a representative set of the particles they pick, that can be helpful. Any other questions, comments? Anyone else have a different experience with picking?
Those bad particles will be removed by 2D, right?
Well, that's the thing, is what does 2D pick? Does anyone know how the 2D algorithm works?
From 2D you will see the good particles and bad particles, right?
Well, so what is your idea of a good particle?
You will see the drop for example, I work with ribosome, and in some cases we try to ribosome with small and large small subunit, and there's a line between, and also we can recognize the top wheel and bottom wheel, so we can
remove everything that's not that. But what if you have a large conformational change and only 5% of your particles have that conformational change in there, and you do 2D classification? Do you think you'll still be able to see those nice views if they're only a small fraction of the data set?
Yeah. No.
That's the issue with 2D classification is everything that's very highly abundant, you'll get back. Anything that's low abundance, and some particles have very preferred views, so you're only going to get top side and maybe one other view, and that can be fine. Other proteins, you have essentially a totally random set of views, and sometimes there's only a small fraction that have one particular view, and by 2D classification, because you're sampling over a fairly wide view and forcing it together, you can end up throwing those particles away, even if they're great particles. And so, same with 3D classification, you want to see how much you're throwing away. If you're throwing away 10%, then most likely that's going to be junk, but maybe not. And so it can be worth, one thing that I always like to do is go back and map all the particles that it thinks are junk onto my raw images and go through and manually look and say, you don't have to do the whole data set, but if you do 20 or 30 images and the things that it's throwing away actually look like good particles to my eye, then I skip 2D classification entirely.
And then you would go more like check what your auto picking was doing. Like if you see, okay, it's really an ice block or an ice particle, you just unpick it.
Right.
Like this, get a cleaner data set this way.
Yeah.
Okay.
Yeah, and I think if you have the time, that's the better way to do it. But then you do introduce bias because sometimes it's hard to tell if you have a small aggregate of 4 or 5 particles or if that's just particles that are aligned similarly but aren't actually touching, and so all should be good particles. And so these are things that sample to sample and project to project you have to be careful of. Most people just automatically do 2D classification, throw away what they call junk, 3D classification, throw away what they call junk. But different reconstruction algorithms deal with this differently. ReliOn, you want to filter down to the smallest data set possible, the most pure, the cleanest data set. Other algorithms, that's not the approach they take. If you take Freeline, what they do is they weight particles based on how well they cross-correlate with your initial model. And so as you go through rounds and rounds of Freeline, what they'll do is the particles that you would call junk, they're just going to downweight them. So in the end, you have the same sort of, you use the same sort of idea that things that don't look the same aren't going to contribute strongly to your reconstruction. But there are 2 different ways to do it, and that's why it can be helpful to try multiple reconstruction algorithms. With ReliOn, you filter down to the smallest subset possible, which you think is the purest, cleanest, most homogeneous. With Freeline, you don't necessarily do that because you can weight particles. It's all done automatically, so there's less bias, which in the end, if you end up with a similar reconstruction, sometimes you can get better from Freeline, sometimes you can get better from ReliOn, sometimes you get better from a subset, sometimes you get better from all. It depends on how heterogeneous your data really is and how well you're able to sort out good from bad. If you're throwing away good with bad, then that'll negatively affect your reconstruction most of the time in the end.
So, if you have a new sample and you have never seen the structure before, so when we pick particles to give factorial alignment, sometimes centering could be a problem. And then there could be good particles, but because they were not centered, it could be like resulting more junk kind of thing, and it could be removed. So, how do you prevent that?
So that's why I will go back and I'll map back the particles to the raw images. And oftentimes you'll see that if you do auto-picking, say my cup is the particle, but auto-picking picked here because you have another one that's right next to it. And if the size difference between particles is not quite right for that, it'll pick one or the other but not both. Then I'll go through back to the initial particles, delete that and pick this one and pick the nearby one.
So, you need to go by each image?
If it seems to be a significant population that is not able to align well, then I will. If you end up with a 3-angstrom map without doing that, then it may not be necessary. But if you're stuck on the edge, you got a 4.1, 4.2, and you know a little bit better can make a difference, then sometimes that can be 10, 20, 25% more particles that you can get. And that may make a difference, particularly if you're really stuck for data collection time. If you get one data collection time every 2-3 months, then it can be a good use of your time over the interim time.
So you need to go back where we have to manually pick particles again?
So after you've done auto picking, you can manually alter that set. So with Reliant, you can display it. With System, you can display it. And then if you can clearly see that it's mispicking or it's picking off, then you delete it and then repick where you think it belongs. Another way is with Reliant is you can extract recentered and then use those to redisplay, and sometimes that'll do it. If your particle initial picking is way off, then sometimes it's kind of stochastic how well it'll be able to center it. And so you can recenter a couple times and that can get out of that hole. Does that make sense? Any other questions? Because this is to me the most important part, is how you get your data set and how you manipulate it. This is where I spend almost all of my time, and I work with my people to try to come up with the optimal ways and to test different ways, because just because someone's had an algorithm that worked really well for one data set, it's not necessarily going to work for the same data set, even if they're very similar proteins. Because they all behave differently, and even one data collection set to another can behave a little bit differently.
So for example, if you have these in the unmasked classification, you have 3 classes with over 25%. Why didn't they pick the first one? Process the first one further.
They might have, and it might have just been lower resolution, or it could have been a smear of these two. These are things that it's always, if I had that, I would do exactly what you said. I would do a reconstruction of anything over about 5%, see if they're meaningful. It may be that it's slightly different. From here it looks like this whole domain is gone. See right there versus here, here, and here?
Yeah.
This domain just seems to be entirely missing in that 25% particle population.
But would it make sense to combine all the bad particles and see if we can get still some good particles which are just classified in this?
Yeah. And they might have done that, but in the end, the point they were trying to make was that it can be closed or open. And so no matter what, you can go through all of these things, but in the end you want to try to make a biologically meaningful conclusion. And so it's important to not just sort and sort and sort for the sake of sorting, but sort so that you can make a hypothesis that you can test in an orthogonal system. Because structure in the end isn't the data. The structure is a way to generate a hypothesis that you can test with biochemistry, with genetics, whatever it is that works well for your system. Structure is a tool to generate hypotheses that you can then test. And so sometimes going down this wormhole can be really helpful because you can learn something new. Other times you're classifying for the sake of classifying. And this is where the cutoff can be really hard, because you don't know at the beginning how much are we going to be able to pull out of a data set. And so you want to sometimes work and work and work, but you don't want to sort and sort and sort for just trying to get 0.1 angstrom more resolution, or this one domain I can't quite see a few side chains. Is that going to answer something that's important to the project that you're trying to do? So this is just a little bit more multi-classification, using masking, and then background subtraction. These are the approaches that they used there. And then the thing that I wanted to end on, and I think we've started this conversation, is how can you use heterogeneity to better understand your specimen? And this goes back to your question, is can you classify the fraction of particles that belong to a particular state in different ligand states, different pH, whatever it may be that you think might be able to manipulate your particle population? But what other sorts of information can you get from heterogeneity? Any ideas? Do you have any examples from your previous work, Ed? I know you worked with some pretty flexible molecules at one point.
Very flexible, but there's kinetics, basically. Different ligands will shift things to different states. That can be meaningful, especially when you have a lot of different libraries of ligands, because you can have multiple conformations. Not all of them are active, and some of them are very important for activation.
So with your heterogeneous sample, how many of them do you think are meaningful? How many of them do you think are artifacts? And have you thought about ways that you can use that heterogeneity to help you answer the problem that you're trying to address?
It's hard to tell. So the negative state data sets, this isn't terribly high resolution. The molecules are incredibly flexible. They're long, sort of rod-like, and they tend to just sort of flop around quite a bit.
Sounds like your stuff, Ed.
Everything I work on.
But so heterogeneity is sort of like the center of this sort of biological question that we were working on. So these proteins exist as homodimers, and there's one really strong dimerization interface that shows up in crystal structures and biochemical experiments. But we think that there are other interactions that aren't quite as strong, that we haven't been able to see biochemically or in crystallography. So we're still early on trying to actually build models of this, but just looking at the raw data, we think that the heterogeneity exists because there are some of these sort of weak dimerization interfaces elsewhere in the protein, and these sort of rod-like things that tend to sort of flop around like this.
And does that have correlations with biochemistry or genetics or any other information that you have previously about the system?
That's our sort of hypothesis. Once we get the model, we're able to sort of tease out this heterogeneity a little bit more. We'll be able to test that. Yeah, it's hard to describe without going into a lot of the function of the proteins, which
That's often the case, but I'm glad that it's able to help. And yeah.
Just have a question about trying to maybe understand protein kinetics based on cryo-EM data, because it seems like it would be hard to, let's say you had some long, I don't know, flexible protein, and you see this range of conformations in one data collection, but then, how could you really control for things like ice thickness and things like this, like really kind of precisely to really be able to understand this in a really, accurate way, because who knows, next time, maybe the next data collection you've added a ligand and you expect it to now only go through 2/3 of the conformations, but this time your ice thickness is thinner, so you're actually eliminating it somehow, or so what do you think about that? Wouldn't a person need to be very cautious in interpreting the kind of dynamic range of protein movement based on, one data set or even a few data sets, because of the, inherent randomness of some of these, sample preparation and things I know they're trying to attack here at the center, right? Just trying to more reproducibly make grids and things like this. So do you have any thoughts on that?
Does anybody else have any from their experiences or papers they've read or just conversations with people? Because those are really valid questions that freezing is still too stochastic of a process to be able to say exactly that statistics that you derive from one sample are going to apply to others. And so in that case, it may be helpful to, rather than collect one huge data set from a grid, make 4 grids at 4 different times and 4 different preparations and use those as independent points, compare between them. So rather than collecting 2,000, 3,000 images from one grid, collect 500 from a couple, see what the internal variation is, and then see how that correlates to variation between specimen. Because if your internal variation is higher than the variation between specimen, it's going to be really hard to make a correlation. The other way is to try to do a single molecule assay through an orthogonal approach. Some proteins, there's great single molecule biophysics that have been done, and that might give you a way to test or a way to generate a hypothesis that you could then test with EM. And so using 2 different approaches. The other issue is we're looking at things somewhere between 0 and liquid nitrogen temperature. We don't know the actual temperature of the specimen when it freezes. It's not 0, it's not liquid nitrogen temperature, it's somewhere along that trajectory. And as you said, ice thickness. The thicker the ice, the different rate cooling is going to be at different layers. In the very center, it's going to be frozen differently than at the edge. If it's a very thin specimen, then it should freeze much more uniformly. And these are all questions that haven't been well characterized, and some of the work the centers done here looking at specimen with tomography, sometimes you have your protein all stuck at the bottom or the top, sometimes it's in the middle, sometimes it's very random, and these could all influence the biophysics of how the specimen reacts, because we know the temperature will shift binding equilibria, will shift equilibrium between multiple states, but we don't have enough data points to even know what is the temperature. There's no good reporter to say, okay, we froze this and it's actually minus 100 C. This other sample's minus 120. And no one's done that reproducibly because there's not a good way to do it yet. But that doesn't mean it can't be done.
Last slide.
That is the last slide.
Okay.
Benefits of heterogeneity. How to make it not your enemy.
Right.
Normally it is, but sometimes you can get, like in your case, you can learn from it. Any other questions?
No, if not, let's thank Rich for the lecture. Again, operational. Keep on talking. So again, next week we won't have a seminar here. It'll be rescheduled for April 5th. So it'll be a little bit out of order in terms of the 2D crystallography. We'll get the intro after you've already received chemical reconstruction.
So next Monday's class will have to cancel.
It'll be moved. The March 27th class is moved to April 5th. If you cannot attend, let us know. We will live stream that one, as always. Yes, correct. Okay. As always, if you have questions, you can always email emg@nysbc.org, and we'll see you next time.